Welcome to Chapter 15. In Chapter 15, we're going to follow the historical narrative where we talked about Mendel now. We have this idea of kind of that genes exist. We have kind of a rough idea of how they work in terms of complete dominance and incomplete dominance and some of these other characteristics of when they're expressed and when they're not. But at this point, we still don't know what genes are. Like if you look inside a cell, where is a gene? And so one of the first steps we have in this process of figuring out exactly what a gene is was the connections they made between meiosis and chromosomes, and ultimately what Mendel came up with, the law of segregation, law of independent assortment. And so they were able to figure out that somehow these genes were on chromosomes. So chapter 15 is going to focus on chromosomes. If you're wondering why there's a picture here of a redhead or a ginger, if you will, with very pale skin, we'll discuss this a little bit later, but there is this idea that oftentimes genes can be on the same chromosome, and so sometimes they'll be inherited in pairs. So this will kind of go against the idea of independent assortment that Mendel had talked about. We do occasionally see situations where it appears that two genes will be linked, that they'll kind of come together as a package deal. And so one of those pairs in humans appears to be pale skin and red hair. And so that's why if you're a redhead, there's a good chance that you should put on some sunscreen because you're a very good candidate for skin cancer. Now, chromosomal inheritance, so we've got this idea now that genes are going to have specific loci, or locus if it's singular, which just means a specific spot on a chromosome where they exist. So we're still not quite sure what they're made of, but if we have our chromosome, there's going to be like a specific section of that chromosome that's going to be a gene. And it's going to be the same section on the same chromosome for every member of a specific species. And so for us, we have a gene that might be at a certain spot on chromosome 1, and we'll have another gene that's further down chromosome 1, etc. Chromosome 4 is going to have a bunch of different genes, but at specific locations. Now, the other thing we have is that we've realized that chromosomes, and this is part of how we knew they were involved, they will undergo segregation. So this is the separation part, where we go from being 2N, and when we're done with meiosis, they're officially 1N. So if you remember, segregation is this idea that you have two of each trait, or two alleles, and when you make a gamete, you pass on one of those two. And so that's the idea of segregation, and that melds perfectly with meiosis. The other idea is independent assortment, where the traits that we have, the genes that we have, I should say, ultimately separate independent of each other. So what you get for one gene does not affect what you get for a second gene. They're not connected. So these guys are ultimately split. And so we call this independent assortment, and we see this as well, where you can get mom's version of one gene and dad's version of another gene. You can get mom's chromosome 1, and yet you can get dad's chromosome 2. You can get mom's chromosome 3 and mom's chromosome 4 and dad's chromosome 5, on and on and on. And so because you don't have to grab, just because you grab the first chromosome of mom's, you don't have to grab the rest that are mom's. We don't see things that tend to be packaged together. So most traits will not be packaged together. Most genes will not be packaged together. You can ultimately get whatever you want for hair color, and then later on you can get whatever you want for eye color. It doesn't have to be connected. Now, there will be some instances where this is not going to be true. And we're going to talk about Morgan. He's just the scientist that really came up with a lot of this genetic research that first involved chromosomes. And so he started to notice some anomalies. So when we looked at Mendel's work, we kind of expected this normal segregation, this normal independent assortment. And so he was working with fruit flies, and he happened to notice that when he crossed these guys that he thought were heterozygous, so they have a red eye allele and a white eye allele, he assumed the male had the same. It was a male with red eyes. That's dominant. It's also called the wild type or the normal. And so he had gotten ones that were supposed to be heterozygous for this white eye condition, which is called a mutant phenotype, or just kind of an odd one, a rarer one. And so when he crossed these heterozygotes, he thought, he expected to see this 3-to-1 ratio, which we've talked about plenty, and so ultimately he did. He got this 3-to-1 ratio. But something was very wrong. So when he looked at this, he realized that a full 50% of the males had white eyes, but 0% of the females had white eyes. All the females had red eyes. And this made no sense, because before, whenever you had this, the gender didn't matter. 25% of them had white eyes is what he expected. That's what happened with every other monohybrid cross they'd done. And gender didn't matter. But suddenly gender was a huge deal because only males were affected, and a lot of males were affected, half versus none of the females. And so that started him to look at, well, maybe there's something different about the chromosomes for males and females. And so he came up with the fly system, which also happens to be shared by humans. So in this system, a female is going to be XX. A male is going to be XY. And we've realized now that there's a lot of traits that are specifically on this X chromosome. The Y chromosome is pretty small. It doesn't have a whole lot of genes. It can't have anything important because only males have it. So if you had where, something like blood clotting was on the Y chromosome, all females would die from an inability to clot blood. That would not be good for the species. So all the important stuff's on the X chromosome. The difficulty here that makes it where sex linkage is what we call this, genes that are on the sex chromosomes, the difficulty here is these guys tend to target males. So if we kind of draw our little target here, so this would be, let's see if I can squeeze it in there. Okay, it looks terrible, but that would be males. So for males, they only get one X. So imagine if we've got these coins, and I say if you get a coin that says, get out of jail free, you're fine. If you get the other coin, we're going to kill you. And so I let you reach in, and if you're a female, I let you grab twice because they get two X's. So they might pull one out that says, uh-oh, you're screwed. But then they get to reach back in, and they have another chance to pull out one that says, you're good. And as long as they get a single good allele, a single good chip, they're fine. For males, they get to reach in once. They only have one X. If they grab the bad chip, they're done. And so in this case, with these X-linked disorders, you're going to see that the mother is going to be the only one that's giving the males an X. And so if she happens to be a carrier, if she's a heterozygote, if she gives them, which half the time she will, the bum allele, the bad allele, the male gets it. He has nothing else to get that will save him. The father can't give him an X if he's going to be male to help him out. So even if the dad has a healthy X, doesn't matter. Whereas the daughter, if the dad's healthy, will always be fine because the dad will give her that healthy, and we're talking about recessive conditions, so the dad will give her the dominant healthy allele. So even if the mom gave her a bad one, she's still good. She's a carrier. She could pass it on to her sons now, but she's not likely to be affected. We'll watch a thing in class about sex determination because this is not the only system, the XX and XY system. But we tend to talk about it the most just because a lot of stuff does use it, especially us. And we tend to think a lot about ourselves, so we do tend to do this. We'll talk about some disorders for humans a bit later, so hold on. Now, the other thing that he came up with was this idea of linkage, which is that two genes can ultimately be on the same chromosome. So if I've got this chromosome, I can have essentially gene A that might be here and gene B that might be here, and you can even have more where there could be a C and a D. And so when you do get this chromosome, via meiosis, you tend to get all these genes together. And so, for instance, if this one tends to have tall and B could be like brown hair, and so you would tend to be tall with brown hair. Maybe the other chromosome has short and blonde hair. So you'd expect to see a lot of people are tall with brown hair or short with blonde hair. They don't appear to be independently assorting because they're not. They're not on different chromosomes. Chromosomes independently assort. Genes independently assort if they're on different chromosomes because the chromosomes are independently assorting. If they're on the same chromosome, they're a package deal. And so you'll be able to tell that there's linkage because you'll have a really large amount of certain phenotypes. And so you might see where in a particular thing you have like 90% of them, and we'll break this down, so say 45% are going to be tall and brown-haired, and you'll have maybe another 45% then that are going to be short and blonde hair. I'm not trying to be cruel to blondes here. And then you've got 5% that are kind of the mixes. So 5% might be tall and blonde, and another 5% might be short and brown. Now, when we look at this, that means that 10% of them are different than the parents. 10% of them are different than what we had before. And so we'll call this, later on you'll see the recombination frequency, and it tells us essentially how close things are on the same chromosome. The reason that how close they are is important is because when you're going through this process of linkage, you can have crossing over occur. And when crossing over occurs, it's going to allow us to break linkage. So if we've got these two alleles, or two genes, so in this case we have gene A and gene B, if crossing over occurs in between them, it can actually separate them. So we can essentially get where crossing over between these two chromosomes will give me big A, big B, whereas prior it was big A, little b. And I can also get what crossing over gives me little a, little b, instead of little a, big B. And so the further apart two genes are on the same chromosome, the more likely we are that crossing over will occur between them. So it's kind of like taking a big long rope, stretching it across a room, tying two flags on it, blindfolding somebody, giving them scissors, this is going to be a great idea, and then having them just kind of randomly cut it in one spot. If those two flags are really far apart, there's a decent chance, assuming that you manage to cut the rope and not someone's arm, that you will be able to cut in between them. And we'll have recombination, this mixing up, occur pretty frequently. And so you might have something where it's like 30% of the time you get, tall and brown, and 30% of the time you get short and blonde, but then we have a high amount of recombination, 40%, that's a lot. So these guys are far apart. Whereas if they're right next to each other, the chances of you blindly with like 20 feet of rope snipping in that 1 inch between them is very low. So you could have something where it's like 49% one, 49% the other, and there's only like 2% overall recombinance versus the 40. And so as we look at stuff, you can get a sense for kind of where things are at. And we use this data, we use these recombination frequencies where we add up the two recombinants, the two oddball phenotype combinations, and we'll use those to make what we call linkage maps, which is where we just say this would be 40 map units. It's just kind of an imaginary thing we came up with, where a map unit is equal to 1% recombination. So this one would be 2 map units apart. So if we know there's enough alleles, enough genes, that are on a chromosome, so we've got A, B, C, D, I can start comparing recombination frequencies of how often does A and D recombine? How about A and C? How about A and B? How about B and C? So I can get all these different recombination frequencies, and so maybe I find out that between A and B it's only a 5% amount of recombinance, so 5 map units. Maybe between B and C it's 10. And that means between A and C I'd expect to find 15. So if we get enough pieces of data, we can ultimately construct exactly where these genes are at. I shouldn't say exactly, relatively where these genes are at versus each other. And so I don't know exactly which spot on that chromosome. There are chromosomal maps that do that, where they look at specific spots, physical locations. But this at least gives us a good idea of how far apart genes are on the chromosome, and ultimately like the order. So I could figure out, that A's obviously on one end, D's on the other end, and B and C are in between, and they're closer to A. I could figure all that out if I have enough data points, such as A and D, let's say, is going to be like 30. And so I could figure this out. Just knowing those numbers, I can construct this linkage map. And so this linkage map gives me a pretty good idea of how far apart things are, how often crossing over occurs, and how often then we'd expect it to be paired up. Now in human sex linkage, there's several kind of cool things that we have disorder-wise. Cool, I use as interesting. First of all, I just love this because the gene that's on the Y chromosome that makes you male, that ultimately codes for you to develop testes, etc., is the SRY gene, which I always call the SARI gene. So the SARI gene is what makes you male. And then other disorders, and you'll see these are all essentially going to be recessive disorders, which makes sense. We've said that's the norm. We have color blindness, so some types of color blindness are going to be X-linked. And this is why males are 90+% of the time the ones who are colorblind. Duchenne's muscular dystrophy is only males are affected, because people that get Duchenne's muscular dystrophy are either dead or incapacitated by the time they would reach an age where they could sexually reproduce. And so people with Duchenne's do not sexually reproduce, which means they cannot pass it on. Because for a daughter to get Duchenne's, the father has to have it, because the daughter has to get 2 bad alleles. So one from the dad, one from the mom. Now if you're a mother that's a carrier, there's a 50% chance any of your sons will have it. So this is one that can really suck if you have it, or if you're a carrier, I should say, as a female, because you really have to watch it. If you wanted to have boys, there's a 50% chance that they would come down with this, and it's not a particularly nice death nor necessarily a very comfortable life. And then hemophilia, there's several types of hemophilia. Some of them are going to be X-linked recessive as well. And so this is why hemophilia tends, yet again, to affect males. So the general rule here is if it's sex-linked, males tend to be affected the most, because if it's Y-linked, always males are affected, because they're the only ones with a Y chromosome. If it's X-linked, males are normally affected, because they only have one X. And so because things are recessive, almost always, it makes it where it's very easy for them to get one bad allele, and then they're just screwed. It also is one of the few things where you're allowed to blame your mom, because ultimately, if you get a sex-linked trait as a male, you got your only X from mom, so that one's kind of legitimate. She didn't do it intentionally, but that one is something where ultimately just your mom did it to you. Now, bar bodies, this is kind of a cool idea where when we look at our sex chromosomes, we only need one X, right? We've talked about this idea that males survive and they have one X, females have two X's, but they don't have to have the second X. And so what most organisms do is they will take the second or any additional X's they have. You'll see there are some screw-ups where you get like 3 X's, and so you'll take anything beyond the 1, so in this case the second and third, and you will condense them down and kind of leave them as chromosomes. And this is cool because while they're a chromosome, while they're condensed, they don't really work well, they don't really turn on well, and so this gives us some cool patterns, but they only exist in females because only females have 2 X's. And so that's like the tortoiseshell cat where they have ultimately orange hairs and they've got black hairs. So they look like this kind of codominant situation, but in reality what's happening is some of them are shutting down the X that has the orange allele, and some of them are shutting down an X that has the black allele. So depending on which one got shut down and condensed, you get either orange hair or black hair. And so you get this tortoiseshell pattern. And so this one is found on female cats, and it's not due to codominance in this case, although it's very similar in overall phenotype. This one's actually due to these bar bodies and this idea of X inactivation. All right, I'm going to wrap this one up here, and we'll finish this up a little bit later. Hope you guys enjoyed it, and I'll see you soon.